gone for a postdoc in China at Nanjing University. And after I got the position of associate professor in Shanghai. And he sent me a message 2 weeks ago because he wanted to see the microbial collection of the museum collection and sample and use it for his research. And I said, oh, you are in Berlin, they just come and see the collection on live and try to make a talk if you can, if you are happy to do that. Just we welcome him and thank you very much for coming and I enjoy talking.
Yeah, thank you. And thank you for the very nice introduction. So today I'm going to talk. I have to put something here for Zoom. It should work. Yeah, I want to talk a little bit about the project that I'm running right now. So from the Ediacaran, I moved a little bit further back in time, be interested in the Archean and the evolution of the earliest life. And of course, if you work with the earliest life on Earth, it is microbial, and microbial life brings you to stromatolites. And so I drafted a little bit of a research project over the last 3 years that I call the Stromatolite Geochemical Archive. I'm going to talk how we use trace elements and novel isotope systematics in stromatolites for reconstructing these microbial habitats through the time. First of all, I want to give a little bit of a background how I actually ended up in China. You gave a nice introduction already. So I did my diploma in University of Bonn in Germany, where I first worked with radiogenic isotope systematics on volcanoes, so igneous rocks, high temperature geochemistry. Oh, more people. Welcome. Fine. And then I did a PhD at Free University of Berlin, as you mentioned, and that's when I first got into touch with China. So it was a kind of a Sino-German research collaboration that we had there, and we had a lot of field work in South China, where I sampled these rocks from the Neoproterozoic, and I fell so much in love with the countryside there and the food there that I thought, well, it's a good idea to go there and work there in the future. So I have constant access to these fantastic outcrops and the good food. And that's how I became a postdoc at Nanjing University in China from 2016 to 2018, working with the same people that I had collaborated throughout my PhD already with. And then in 2019, I thought, okay, enough of the small cities. Nanjing only has 10 million inhabitants. I want something bigger. So I went to Shanghai, which has 25 million people, and there started to work as an associate researcher first, and now associate professor in the School of Ocean and Earth Sciences, and there especially in the State Key Laboratory of Marine Geology. So from the Neoproterozoic, I went into modern marine environments because I thought I had to better understand what's happening in the modern oceans to then extrapolate this knowledge into the past. I built up a small work group there where I'm a junior leader of the so-called Tongji carbonate geochemistry group. I have one postdoc, 2 PhD students, and a master student who graduated this year. And the aim of our group is to develop novel stable isotope and innovative trace elements and dating methods in carbonates. We use multi-collector ICP-MS. We also do a little bit of laser ablation work on carbonates. Also, I'm teaching the graduate student course, Principles and Applications in Polyenvironmental Geochemistry. What is the motivation of all of my research? So let's dive a little bit into that. I like very much this publication from 2008 by Ariel Ambar in Science, where he tried to actually scope what would have been the evolution of element distribution in the oceans over time. Do we have, I can use this. So some of you may be familiar with this plot here, which was published first time by Lyons, or Canfield did that as well, but it actually shows how the oxygen concentration in our atmosphere changed over Earth's history. So you see here on the x-axis basically the time of the geological timescale, with today being on the right-hand side and 4.5 billion years ago on the left-hand side. And you see that the oxygen levels in our atmosphere have never been constant as they are nowadays. In fact, they have been very, very low throughout the geological period that we call the Archean, which basically spans the first 2 billion years of Earth history. And at that time, we had extreme anoxic, meaning oxygen deficient atmosphere and also very low oxygen dissolved in our oceans. And there was a period of time around 2.5 billion years ago when that rapidly changed. We call this the Great Oxygenation Event. And that basically was proven by some sulfur isotope work, not going too much into detail, but researchers found out that there are new geochemical fingerprints in the rocks that told them, suddenly we have more oxygen available in the atmosphere, in the rock record, and so also people turned into the marine rock record and were trying to figure out whether or not they could find hints for the change of oxygen in the oceans. And so Ariel Amber then said, like, well, if we have changing oxygen conditions in the oceans, that must have influence on the availability of certain trace metals, especially those that are redox sensitive. And some of these redox sensitive elements may have been in dissolved form extremely enriched throughout these anoxic Archean oceans, such as iron or manganese, but may have dropped to lower levels when the oceans became more oxidized, because then these metals tend to form oxide phases. And these oxide phases are immobile, they are not dissolved, and some of you may have seen these as these kind of very, very small, sorry if you cannot see this, is the kind of a manganese or iron oxide nodules that you can find on the modern deep ocean floor. And this is basically an oxide that is formed out of trace metals and iron and manganese. So they are in solid form, they are not dissolved. Yeah? So and that is why these concentrations drastically dropped through the evolution of our oceans in deep time. Other elements, however, such as molybdenum, or let's look here at zinc or copper, they went up throughout the evolution of our oceans. And that is because under oxidizing conditions, they are more common in dissolved form. Whereas under anoxic or even euxinic conditions, that is when you have free hydrogen sulfide present, they are locked in a kind of mineral called sulfide. So why is all of this interesting? Because these trace elements hold crucial biologic essential functions, because they can act as cofactors in enzymes. So the evolution of life on Earth may be, or in the oceans, may be directly linked to the evolution of the availability of these trace elements through time. My question is it able then, or are we able then to trace the abundance of these elements through deep time? Some researchers have therefore focused on special kind of rock called black shale in the past. Those are very organic matter-rich rocks, marine sediments that are formed in anoxic basins, and there they had looked closely for these pyrites, for these specific type of minerals, and tried to trace the availability of these elements through deep time. They came up with these beautiful elemental curves in these sulfides, and they found that there were times where the oceans were rich in these elements. And they said these are nutrient-rich oceans. So you see where you have these peaks, for example, in the trace element selenium, which is a very important nutrient also for us. And then there were times where the oceans were deficient. And they said that this is also correlating with mass extinction events or crises in bioproductivity. Well, my problem that I always had with these kind of archives was that they are very specific. They are in deep sea, they are in anoxic conditions, they are always these kind of restricted anoxic basins. And I, coming from another background, the carbonate background, wanted to know what's actually happening in the surface oceans, where oxygen much earlier arrived. And so I wanted to think whether we cannot use, instead of these sulfides and these black shales, carbonate formations to trace the availability of these trace elements through deep time. My background on carbonates is that I started to work throughout my PhD on the carbonates from the Ediacaran, and I developed some special methods to actually trace whether or not a carbonate is pristine. So I used some kind of petrographic methods where I use microscopes to actually look into the carbonates, and SEM, like spectral kind of high resolution microscope, and also trace element and radiogenic isotope methods to figure out whether or not these are good archives. Then later I developed throughout my postdoc times a kind of sequential leaching method where you attack these carbonates in that way that you only dissolve the carbonate minerals and not some impurities that may arise from other minerals, such as silicates, that could be clay minerals, or that could be feldspar or oxides that you don't want to attack. So we actually developed a method where you use different acids with different polarity so that you only attack one specific mineral's phase, one after another. And then throughout my current research, I focus on these so-called stromatolites. And that's what the rest of the talk is going to be about, because I believe I have found now the right archive to look for the evolution of the trace elements through the time, and even some isotope species through the time by looking at this particular kind of rock. But I think I first have to explain to you what are stromatolites. Here's a very fantastic collection of stromatolites or microbiolites in the museum. So some of you may have seen samples here already, but for those who are not familiar, I want to give a little bit of an introduction. Here we see some field photographs for stromatolites. You see stromatolites are very strange objects. They are some kind of fossils actually. And these are rocks mostly formed of carbonates, but there are also other minerals that form these rocks, and they are actually formed by the interaction of organosedimentary structures formed by incidental induction of mineral precipitation on microbial biofilms. What does that mean? So here on the left side, we see a very nice picture of a modern microbial mat. We see a stack of different microbial communities stacked upon each other. On top, you see a very greenish, gluey kind of substance, this kind of substance called EPS, extrapolymeric substance. And this is actually a substance that is excreted from the typical community of bacteria that also involve cyanobacteria or other phototrophic organisms. Phototrophic organism means it's using sunlight as a main energy source to do or perform photosynthesis, where you can split H2O molecules to gain energy using the energy of the sun. And when you go a little bit deeper in this microbial stack, you see that the color changes. Yeah? At the top screen, that's coming from the cyanobacteria. They do the photosynthesis just as plants do nowadays. But if you go a little bit deeper, you see the color change to like brownish, reddish, and that must come obviously from a different kind of bacteria or microbes. In these stacks, you not only find bacteria, you also find another kind of microbe called archaea. You also can find some eukaryotes. You can even find viruses in these stacks. But what we are mostly interested in is actually what all of these microbes do together. They together create a special environment with increased alkalinity, and in this environment, it happens to precipitate these white layers here. And these white layers are carbonate minerals. The carbonate minerals are in direct exchange with the water in which these microbial mats grow. And when they grow in these environments, they actually take up nutrients, so that is trace metals dissolved from the ambient waters, and incorporate them into the microbes. When they die, eventually they release it again into the pore waters. So you have uptake and metal recycling happening within these microbial stacks. And whenever there's these carbonates being formed within, they actually record the ambient water composition. And that's what we are interested in. Of course, they're also very beautiful to look at. Let's focus again here on those pictures on the right side. You find them in the field, they look amazing. But what I'm mostly interested in is what they hold as a key information on these elemental evolution through deep time. Why is that? Because stromatolites exist throughout deep time. From the Archean eon, which dates back up to 4 billion years before today, till modern times. Here you see an example from the oldest non-disputed biological origin stromatolites from the 3.45 billion year old Stromatolite Hohl formation in Western Australia. Here you see a photograph that was taken underwater off the coast of Bahamas. So from 3.45 billion years to today, you find stromatolites. And we have been doing a couple of analyses on these typical rock types and wrote some papers on it, and we believe that these stromatolites can be introduced as a very new geochemical archive to study the geochemical conditions and the geodynamic evolution of shallow marine environments through deep time. And this is the key point here. Shallow marine environments, because they need sunlight. These microbial activities performed at least at the top phototrophy, so that means photosynthesis, and therefore they need sunlight. Sunlight can only penetrate the seawater or the lake water up to, let's say, 10, 20, maximum 30 meters depth, cannot go much deeper to find these stromatolites. So this is a very nice archive that helps us to explain or study the changes of trace element availability through deep time in the surface waters. Unlike these black shells I talked before about, because they represent deep marine environments or deeper marine environments. And why is that interesting? Because if we are able to date back up to 3.5 billion years with the stromatolite archive, then we can actually see how the evolution of certain microbial communities that build these microbial stacks changed the seawater composition or the ambient water composition. And you see that at the beginning, 3.5 billion years ago, we are not actually sure what was the microbial community at that time. There may have been, I guess, there's not much of debate about that anymore, methanogenesis and nitrogen fixation dating back about 3.5 billion years ago. But we are not absolutely sure when the first photosynthetic organisms showed up. Oxygenic cyanobacteria most likely appeared at this so-called great oxygenation event that I mentioned earlier to you. This is when the oxygen concentration in the atmosphere suddenly skyrocketed. And that is because we had a new kind of energy source that could be harvested via photosynthesis. And the side product was oxygen, right? So these, whereas the methanogenic organisms before were harvesting on methane, then later on the phototrophic organisms, they were harvesting the sunlight as an energy source, and they were cooling oxygen. So their side product, oxygen, was toxifying the environment for the methanogens at that time, which died out. But they were also toxifying themselves because they were eventually oxidizing iron and other crucial radiosensitive metals in the seawater, which they used as cofactors or nutrients for themselves. And they formed a rock type called banded iron formation, which was the oxidation of iron and forming massive iron deposits in the oceans, eventually causing a kind of a early extinction event. It sounds familiar to what's happening to us nowadays, right? We are harvesting a new energy source, fossil energy, and we are extracting a lot of CO2 and we are toxifying ourselves. That's what eventually happened when the first cyanobacteria showed up on planet Earth. So if I'm able to actually then trace this evolution of the different metabolisms through time, I get an idea on how these metabolisms change the environment. Here, by the way, we see a very nice photograph from Tanya Bosak, where you can see how these microbial stacks, especially the photosynthesis in the top layer of such a microbial stack, actually produces oxygen. You see a beautiful oxygen bubble here coming out of these cyanobacteria. So there's still a lot of open questions. For example, how and when exactly did these diverse microbial communities evolve on Earth? How does microbial life cycle bioessential elements in the oceans, and can we trace the nutrient availability in the rock record? And how did the interaction of the microbes with the environment shape the early habitats? And can novel stable isotope help us to trace the extent and the timing of these events? And also we could probably answer questions that are big in the news nowadays. For example, there are some even older stromatolite-like formations that have been found in Israel and Greenland, and they are dated to 3.7 billion years ago. And some people say, like, well, that looks like a stromatolite as well. They have these kind of tufted and domal structures, but there's a huge dispute whether or not they are actual pyogenic stromatolites. Also, this location here, which is actually a rendering, you see this is a satellite photograph that somebody has filled with blue color to make it look like a lake, because that most likely was a lake in the past, and that is scale crater from Mars. where some of the photographs that have been shot by the rovers show these kind of lake-like sediment deposits, and some people were arguing that eventually, some billion years ago, life was also, or Mars was also a habitable planet, and if we would like to look for microbial rocks and their remnants, we would have to go for places like Gale Crater and look whether or not we find similar structures. So the questions that arise here, how can we finally prove if these really old 3.7 billion year old rocks or these rocks from Mars that eventually will come back to Earth one day when we have sample retrieval campaigns, if they are actually the product of metabolism, microbial metabolism. And I believe that a way for doing this is by using stable metal isotopes. So what we are doing right now is, in order to better understand how microbialites are harvesting nutrients and eventually some isotope fractionation occurring in these rocks, we are building up an archive. As I said before, we have to go through deep time to cover all of Earth's history, but also we cover a lot of locations worldwide where we sampled so far. So here's just a few locations that we went in the past or that we plan to go in the future. And what we want to do basically is to build up then a database that is helping us to identify the long-term trends through this database. And it will also give access then for modelers to test their hypotheses on these kind of data sets. So what we do in the field is this is some field photograph from sampling in Mongolia. We actually go, try to find these ancient stromatolite reefs. We try to chisel out big chunks of stromatolites, cut them, polish them, and then we have these rock slabs where I try to drill them into individual laminae. These are, you can clearly see these microbial stacks that have been lithified, right? And you see layer by layer by layer. We try to drill inside, and I also try to drill into the host rock in which these microbial stacks are embedded to get a kind of a baseline to see what is the difference between the rock just besides it and within these microbial rocks. And then here you see how we actually try to spread this out through deep time, and we have to then access the actual information that is stored in these rocks. So by doing this, we have to keep in mind that microbial rocks or microbialites are actually a combination of different environmental impact on them. So for example, there's the microbial community that acts on the chemistry of these rocks, then the ambient water chemistry that acts on them, and also you sometimes may have a kind of contamination by silicates or like very light clay minerals that are floating in the water that are also trapped on this gooey substance. It's very sticky, so whatever mineral floats by will stick on top of them. And so when you have a calcifying biofilm, then you have eventually a combination of all of these three environmental information. And on top of that, unfortunately, these very old rocks, they undergo a process called diagenesis, where some fluids interact with these rocks and overprint these rocks. And the final product, the so-called microbialite or stromatolite, is a combination of all of these. And we want to make sure to avoid the ambient sediment, because it's information that we don't want to access, and the diagenetic overprint. So in order to get a pristine carbonate geochemical composition, get this kind of archive, we have to use sequential leaching, which only attacks the carbonate and not the ambient sediment, and we have to filter out any samples that have been overprinted by diagenesis using some special methods based on our geochemical analysis on some fluid mobile elements such as oxygen, strontium, etc. And then I can show some preliminary results. I'm not sure how many of you have in a geochemical background. I know some people online have some background. I just want to give a little bit of details here. So one can, for example, use strontium isotopes. That's a radiogenic isotope system. And you can see that most of the stromatolites actually show very high values here that are off what we call the so-called seawater curve. And so radiogenic isotope systems often don't work very well in stromatolites. It's not clear why that is. It could be either because the old age, there's diagenetic overprint, but what's more likely is that they are all often being growing in very restricted environments, very close to continents, and there you have a lot of continental weathering products being brought in, which overprints the strontium isotope composition. But if you look at rare earth element patterns, you see that the rare earth element patterns often overlap with what you see in the global carbonate platforms, which is given here at the blue spots in the background. You see that stromatolites and the global carbonate platforms overlapping in that way that you see, for example, more of these so-called negative serum anomalies in the Proterozoic and then into the Phanerozoic with the highest values, meaning the most negative serum anomalies being in those times when the oceans were more oxidized. So I don't want to go too much into detail on that, but what it actually tells us is that we can use these as archives for certain elements that are not fluid yet. And the elements that I'm most interested in are elements that actually hold some key information as cofactors for enzymatic processes, such as manganese, cadmium, nickel, but also we could look into zinc and we could look into cobalt. And why I choose these elements is because they actually behave as enzymatic catalyzing ions, such as in carbonic anhydrase in the terms of cadmium and zinc, or hydrogenase in the terms of nickel. And also in methanogenesis, nickel plays a crucial role. Also very important for me is that nickel and cadmium and zinc, they hold further information when we look at their isotope compositions, which I will talk a little bit more into detail later on. Here's how I show that cadmium and zinc are very nicely correlated through deep time. Cadmium and zinc are both elements that are incorporated into photosynthetic primary producers because they both are cofactors for this enzyme called carbonic anhydrase. And you see there's a linear correlation between cadmium and zinc through deep time in all of our stromatolite samples, and this has been filtered already for any kind of detrital contamination or diagenetic overprint. You can also see that there were higher values for cadmium and for zinc in the samples with a pinkish color, and those are the older samples coming from the Archean. The size of these data points is given by their aluminium content, and you can also see that while in the Archean we often have a lot of mineral trapping happening, the Proterozoic and Phanerozoic, which is a little bit off term, those are the yellow points here, seem to be more alkalinity dominated. And so we have more of the incorporation here into the carbonates. This is also preliminary data that we did on barium isotopes, but also we did some literature research on a couple of other stable metal isotope systems that was published by us in 2021. This one on barium isotopes is just now under review, and you see how we try to actually figure out how the compositions of these stable isotopes change through deep time. In the terms of barium isotopes, you see that while the barium concentrations in these carbonates, carbonate-associated barium, changed through Earth history from low concentrations in the Archean to higher concentrations in the Phanerozoic, we have a peak in their isotope compositions, meaning more positive values in the Proterozoic, and then a decline again in the Phanerozoic. And that coincides with the actual stromatolite proportion in the habitats through Earth's history. Stromatolite proportion means how many of all the marine rock units that have been described in the literature actually contain stromatolites. And you see there's a clear peak here in the Proterozoic when there was the peak abundance of stromatolites. Why is that? Because these microbial communities had absolutely nothing to fear at that time. But then, with the Phanerozoic and the famous Cambrian explosion, the first metazoans or animals appeared on our planet, and they would love to just graze on these kind of microbial communities because they can't run away. So if I'm an ostracod, I just can go there and like graze on the surface and the microbial map and not do anything. So that's why nowadays we only find microbial communities in very extreme environments. And so we see this actually being represented in the barium isotopic compositions. How much time goes there? Or it's still out? That's okay. Great. I'm not sure how much I should go into detail here, but what I believe is actually happening here is that the decline in barium isotope compositions has to do with the fact that the microbial mats are now retreating into extreme environments, and in these extreme environments, they also cut off from nutrient fluxes. When you cut off from nutrient flux, what you actually have to do is you have to start to recycle nutrients within the mat, within this stem, meaning a dying layer of microbes has to become the nutrient for a new growing layer of microbes. This is how they recycle the compositions, and this is how eventually you can see this in the rock record. I would like to jump directly into this work that we did in rocks from the end of the Proterozoic, Neoproterozoic in Brazil, Paranoa Formation. There we had two different kinds of stromatolites where you can actually nicely see how this is happening, this recycling. So there's one location called Fazenda Funil where you have these beautiful domal or columnar stromatolites, and here you have more domal ones in Sao Gabriel. They have two complete different environments in which they have been precipitated or formed. You can see this again by looking at the so-called rare earth element patterns. Those of you who are familiar with this, just keep in mind a marine signature, something that has been formed in the open ocean, would always show you this kind of a positive or like a positive slope where you have the heavy rare earth elements enriched over the light rare earth elements. And also you find some typical peaks in these patterns. There's one at the beginning at an element called lanthanum, another one here at gadolinium, and there's the enrichment, relative enrichment of yttrium over holmium. And all of this here are data points from individual layers that we have drilled, and this is for comparison on modern seawater. And you see it's very similar, only that this negative peak here is missing. And this negative peak would define us the serum anomaly, the presence of free oxygen. Proterozoic, the oceans were not as oxidized as they are nowadays. In the other locality, where you have also a different morphology, you have more flat patterns. And we believe that these flat patterns actually arise from the fact that they have been formed in a restricted lagoonal environment. Interestingly is we have done cadmium isotopes on these rocks. And the cadmium isotopes are a very new proxy that can actually trace the uptake of cadmium as a nutrient into these microbes for photosynthesis. And when they do this, they fractionate the cadmium isotope pool. For those of you who are not so familiar with stable metal isotopes, I will just briefly walk you through this and try to explain that zero means this is basically how your starting composition would look like. If you start to fractionate isotopes in either way, meaning your microbes take up light isotopes or heavy isotopes, you would have deviation from the zero value. And you see in all our sample points that we actually see deviations in both directions with a spread from minus 3.5 to plus 3.5 epsilon units. Don't need to know in detail what epsilon units mean, but just see that there's a huge spread away from zero. So that means some biological process must have caused this kind of fractionation. Because if there's no biology involved, and if you just measure a kind of igneous rock, then it would always be around zero. You would never deviate that much from the zero line. Interestingly, this deviation, this spread here in the stable metal isotope composition of cadmium, correlates very well with some of these redox-sensitive elements. Cerium, a redox-sensitive element, with the negative values showing you oxidizing end members, and the more positive values showing you reducing or suboxic environments. The same with manganese, which is also redox sensitive, where it means if you have less manganese in your carbonate, manganese has reacted with oxygen to manganese oxide. And if you have more of it incorporated, then that means oxides have been dissolved. So we could easily draw two end members in these microbial rocks, and one end member was oxic and one end member was anox. And that goes along with a nice isotope fractionation. Now you could say, cadmium is a redox sensitive element. Can I use it as a redox proxy? Not really, because it's actually not behaving redox sensitive. What's happening is the cadmium is recycled inside the rocks. And you can see that when you plot the cadmium isotopic composition versus its concentration. You see a linear correlation on the log space, and if you plot this as a kind of fraction of the total cadmium pool, you see that at the beginning, the end number 1, there's a lot of cadmium available, so a lot of cadmium is incorporated, and we believe this is the oxic end member, and then we have a huge fractionation into these phototropic organisms, and they prefer to incorporate the light cadmium. Plus, this is the negative values. This is where the light isotopes are incorporated. The deeper anoxic layers, they don't have access to the cadmium, so they can only repurpose these kind of microbes, what is left over. So they eventually take up very, very lower concentrations of cadmium. So the fraction here is much lower, and they can only take what's left over. And that is the heavy isotopes in the ambient pore waters, because the light isotopes have already been used up. And so you see this kind of, we call it a Rayleigh fractionation curve, where you have this gradual increase in isotopic composition to heavier values while losing your absolute fraction of cadmium in the pore water system. So this is actually showing us that in these samples we had this kind of nutrient use up. And that happened under open marine conditions. We believe here on this kind of shelf, we had the rocks at Fazenda Funil being growing, these stromatolites being growing. There were obviously only sometimes some influx made by upwelling of nutrients, but not enough to sustain a stable income of cadmium. Whereas in the São Gabriel, the hinterland, we had basically restricted environmental conditions, and their riverine input would bring us enough cadmium to have like a stable baseline of cadmium and no fractionation. And you see then this gradient from the outside to the inside of these stromatolites that actually one can trace layer by layer. And that's what I mean with the recycling under restricted environments. This is another example that I like to show. We go even deeper in time. Here we went to the South Africa. Pongola is about 3.0 billion years old stromatolites. They are partly silicified. So we wanted to see whether we could still extract good isotope data. And we did some micro drilling into individual stromatolite layers, but we also did laser ICP-MS in situ trace element mapping here. And we found that the elements of interest, again, these nutrient-like elements, cadmium, nickel, but also barium, were associated into this kind of biointrinsic layering. You always see the accumulation of these elements on top of these stromatolite layers. And so we believe this has to do with the uptake and eventually also the formation of oxides under the production of oxygen, where you have the formation of nickel and cadmium being bound on these surfaces. But with barium, we see more homogeneous distribution. It's not directly bound to these microbial mat surfaces. We could also show that they have nice rare earth element patterns that represent these kind of marine environments, and we could also find negative serum anomalies, so that argues for the presence of free oxygen already before the great oxygenation event, and people call this so-called oxygen oasis. So eventually, even in the Archean, there were some small patches where enough phototropic organisms come together already to perform photosynthesis and create free oxygen. This is why I said I believe that some of these are actually oxides that form on the surfaces of these microbial mats. You see very nicely with the iron that is enriched on top of these small columns, columnar stromatolites. We did then nickel, cadmium, and barium isotope compositions, and again you find this typical behavior where you have some heavy isotope values with the lowest compositions, the lowest concentrations of the respective metal in terms of nickel and cadmium, which both tends to give us information about their biogenicity. because nickel is incorporated into methanogenesis. So this is a kind of a metabolism where methane is harvested or is produced, sorry, and cadmium is the photosynthesis. In barium, we see an opposite behavior, which means we have like heavier isotopic values at higher concentrations, and we believe that this has to do actually with the formation rates of carbonates at that time. But I can't go too much into detail. Who's interested in barium isotopes can later contact me on this. I have a couple more slides on that. But it can be eventually used to theoretically calculate the precipitation rate of the carbonates. And now I would like to sum all of this up. I say our novel metal isotope toolbox can be used as kind of biomarker for microbial metabolism, even in these archean rocks. And this I will just briefly show you what we do on top of it, but I'm not going into detail because we don't have the time here. My team is also working on magnesium isotopes, which can be used as a tracer for continental weathering, for example, but not biologic production, and gives us information about the background of the ambient pore waters. We also do this in situ trace metal maps. I showed you some examples before. We use some innovative method of using matrix matching, very homogenized nanopowder standards to do that. And then we can get these beautiful trace element maps that are fully qualitative and quantitative. And we use this as the first step to analyze where we could actually do some dating using the decay of uranium into lead, and we can actually precisely date the formation rates of formation ages of these stromatolites. So here's an example from a Miocene stromatolite from Austria, and here's an example from Mesoproterozoic stromatolite from China. So what is the aim of our project in future? We want to increase our understanding of microbial metal cycling. We want to accelerate the research in early Earth in deep time and possibly the future sample retrieval from microbial rocks from Mars. I would like to increase scientific collaborations between researchers in Europe and also China, where I work right now. And I would like to establish the online geochemical archive for stromatolites, and I think I'm at the right spot here because we already started to do something similar here. And yeah, that's the idea, to combine all these projects and bring this all together into this so-called geochemical archive. We also do a little bit of outreach. I work with people from the design department in Tongji University at Shanghai. So they did a couple of exhibitions on stromatolites and the beauty of stromatolites and the design of stromatolites. They also did some kind of AI stromatolite painting where they just feed in the words China and stromatolite, and you see how the AI paints something that looks like a mix of a panda bear and a stromatolite. Another researcher did a kind of VR experience where you can bring shark-based stromatolites into your living room. It was a really funny project. And I built this where I used some gray space analysis and turned this into a sound wave so you can listen to the 3 billion-year-old Pongola stromatolites. Let's see if that works. No. I have to get my. You have to. Value, and then you can create this. So that was just playing around a little bit, but the idea is doing some outreach to get people from other departments involved. That was basically playing around with the students from the design department. They were really curious about that. And this is our laboratory. It's directly located on the coast of Shanghai on the East China Sea, behind a kind of a dike, and there we have our sample processing, our multi-collector IMPMC, our laser system, and our quadrupolar IMPMC. So with this, I would like to thank you very much for your attention. And
Thank you, Simon. Very nice and what you guys thought. Do we have any questions? Yes.
I think the Chinese, so I'm going to ask you a question.
Please go ahead.
We talk about Stromatolite.
Yeah.
Like it's a recent in Mars. So like how about the distribution of Stromatolite on Mars? Is there any?
Well, so far not a single one has been found. Yeah, there's like photographs that have been taken by some rovers, and some people fantasize that this could be kind of microbial structures, but we don't have any proof for that. The idea basically stems from, well, the environmental conditions are not so different, or at least back then on Mars, when there was a kind of a bit of an atmosphere and some liquid water present, is not so much different from what we had in early Earth times, right, in the Archaea. So basically everything is there. If you want to have photosynthesis, use the sunlight, it's there. The question is, were there any kind of lipids, like the basic amino acids that you need to build these microbial cells to perform this photosynthesis or methanogenesis or any other kind of metabolism? We don't have proof for that yet, which is why researchers are waiting for the first samples to actually return to Earth. And then the big question is, how do we analyze them? And I think metal stable isotopes are a good proxy to do that, because the fractionation of these isotopes is often happening under metabolic uptake of certain key elements. And this fractionation could otherwise not be explained by other geological processes. So if you want to have a fingerprint for that, use the metal isotopes. Yeah, please.
Subject. Like the picture that you showed us about this structure on Mars, and like the most possible hypothesis wouldn't be like kind of cementary structure, like object foundation or stuff like that. And is it relevant with the environment of the build prior or whatever? Or I mean, it really also looks like other really common cementary.
Absolutely. Right. Yeah.
Like what's most possible than another one, or will I mean regarding the cementary context of the project, would it be more relevant to think that? Or?
Well, that's a very good question. I'm not sure if I can fully answer that. I think the reason why they chose gay craters is because they saw these kind of pans, like river influx into this crater that looks like there's a kind of sediment fan from a river bringing fresh water into that lake, if it was a lake. And then if you're able to get whatever kind of drill core from these lake sediments, maybe those are not the beautiful stromatolites that I showed here, but you would have, if there was life in the sediments itself, some kind of remnants of a microbial community, whether that have been benthic, living on the floor of such a lake, or floating in the water, we don't know. But people are eager to test this for biomarkers or for metal isotopes to see whether or not there was life in that lake.
And the structure and the scale, the spatial scale of that structure, is it relevant with kind of stromatolite communities? I mean, could be a stromatolite community of that extent could be exist. Do you have a local of such temperatures?
Yeah, absolutely. Like when you go to some modern lagoons where you find stromatolites, absolutely. I mean, in scale stromatolites you can find in the tiniest environments, even in some kind of cavity structures. You find stromatolite growing, and we find it in massive lagoons. And back in the days when they were the only form of life on planet Earth, then yeah, they were most likely everywhere at the time in these kind of environments.
You compare them to the one from Canada on the distribution.
Oh, no, I didn't compare them to them. It was more like those are two examples where we would eventually have to test our methods, because the ones they were from Greenland, there's a big discussion because they're so old. They're even older than the ones where we clearly have hints on biologic metabolism being involved. The ones from Greenland, they only have morphologies that look like stromatolites, but nobody has put up like the null hypothesis to prove they are absolutely biogenic, meaning formed under the under the presence of microbial organisms.
What are the scale of that structure?
For the ones from we saw, the whole outcrop is like 1 or 2 meters in scale, and the individual, maybe there is a scale bar on it, the individual, yeah, so you see like the height is maybe like 3-4 centimeters. And yeah, some researchers argue, well, this is kind of up thing that you get when you have these microbial mats building up. But other researchers have shown, well, you can technically also get this if you start to like put something under pressure, you create these kind of wavy structures. So come on, this is like geofantasy. We don't know. We have to test this. So.
No, this is sure that on Mars there is the content, and the figures here are this indication that is a delta, and that is what we call a crater. Now the idea with the stromatolite is that from the orbit you can see some engagement carbonates around the rim, and that might have been at the area force of the visible crater lake. Now, of course, carbonates is not enough to say that
Exactly.
So the next question is how those carbonates like insecure, and then of course getting them back.
Yeah, and that's why I don't know where this perseverance, right?
Yeah, this is perseverance. This is the cartoon of perseverance.
Yeah, but for a couple of weeks or months, it's already like driving to this.
Yeah,
To this rim of the crater.
Into the rim.
Yeah, so once arriving, we start to drill into these formations and then eventually drop the sample or the drill cores there, and they are lying there until they will eventually be picked up in the future retrieval mission, and then maybe no more.
Any other questions?
Ouais, bonsoir.
Okay.
Yeah, I would just puzzle by one thing you said very quickly is about the magnesium isotope. So because you say the magnesium isotopes are really indicating of partners and not of biological processes.
Yes.
But I would have expected at first order that the magnesium
Yes.
would be a marker of
They are in a couple of organisms, in some shells you find that, but we find clear evidence here that there's like no direct incorporation of magnesium into the microbial mats. That's work that's done by my postdoc, and this doesn't work. So what's quite interesting here is that you see that the magnesium isotope composition is directly bound to the magnesium content in the calcite. So this is like low magnesium calcite. And first, when we saw only this plot here, we saw okay, this magnesium isotope composition where this carbonate comes in 2 different stromatolite units. We find opposite behavior in terms of magnesium isotope fractionation. So that was first an idea like does that mean there's a biogenic effect? But then we were able to actually also access the pore waters in which these stromatolites have grown. And we found that the pore waters actually reached the exact same values and the exact same behavior in terms of magnesium concentration versus magnesium isotope composition. So actually the carbonates grow in equilibrium with these magnesium-rich pore waters, just incorporate that signature into the carbonates. So there's no biogenic influence on this. But that doesn't mean that other organisms are not doing it. Just for these kind of microbial communities, that's not happening. But there's other research being done on brachiopods or whatever, and that you find this as actually biogenic influx.
In the case of chromium isotopes, what is the speciation of chromium? Is it organic chromium, or is it oxide, or is it some dissolved in the carbonate?
There is not much of chromium isotope data done yet on the stromatolites, but I quickly can show what's out there. So mostly some modern stuff here.
No, but the one you saw is a correlation on chromium isotope.
Oh, that's cadmium, not chromium.
That's cadmium.
Yeah, for chromium, that is actually it's oxides that then later dissolve. That's where you get this fraction. And for cadmium actually substitutes, because also P+ substitutes for calcium, and it goes into the calcite or carbonate lattice.
Okay, so it's not an organic sediment.
No, it's not found on organics anymore. These rocks are completely devoid of any organic matter anymore, like very, very low TOC. And so what was former bound to the organic matter is now incorporated into the carbonate.
Okay, and it is not oxide.
It is also not an oxide. It could, and that is also common for cadmium, form a sulfide flow. So if you have cadmium being incorporated into sulfide, then there's green oxide, for example, that is cadmium-bearing sulfide. Then you also see a negative fractionation of light isotopes being preferentially incorporated into orthogenically formed sulfide. So you have to make sure that your rocks that you are looking at are not having been formed under sulfidic conditions, because then you are a little bit in doubt whether this fractionation arises from the formation of sulfides or from the incorporation of light cadmium into organic matter.
So we take last question.
Okay,
And after we have a coffee here, and Simon will be here all the afternoon.
Yeah,
So if you want to talk, we will be here.
So you are referring to carbonates.
yeah,
I've never said anything about the mineralogy.
yeah.
And if I look a little bit the literature about all the trace metals and the isotope explanation of these trace metals and carbonates,
Yeah,
there is a difference between calcite and aragonite.
absolutely.
And at the very beginning, you said submarine environment. So I don't know what happened 3.5 billion years ago, but marine environment nowadays mainly precipitates aragonite,
Exactly.
which is not very stable.
Yeah.
So how does this work with the interpretation of your research?
It's a very good question. In terms of nickel isotopes, we have been working a little bit with this because there's actually some experimental research being done on this, and you see that there, as you said, there's different fractionation factors, whether you're forming an aragonite or whether you're forming a calcite. The same is true also for barium isotopes. Marcus said I have done a lot of work on this. Yeah, so that is definitely something that could give us a new perspective on how to use these proxies as well, because it will eventually also, when you show, if you're looking at an aragonite, for example, or if you're looking at a bellerite, which is a barium carbonate, you can use actually isotope value to calculate then back to the precipitation rate, like how fast these carbonates have been formed.
Okay, I am fine with all this, but the question is if you form initially aragonite, and now we are now, and then we go 3.5 million years after, this aragonite will transform to calcite?
Most likely. Yeah.
Or what is the mineralogy in your site? Do you look at it? Mainly calcite.
No, they're also dolomites. Yeah.
Oh, that's even more interesting.
Yeah, exactly. Yeah. So not every stromatolite is alike. So you have a huge variety on the mineralogy when it comes to that, and some of them have also been partly silicified. So of course that puts a lot of extra like open questions on these proxies. And so far we always plot our isotope composition, whether it's independently calculated, like I showed with the magnesium before, and we see for the long-term distribution, so for example, the ones that we have in this Sandrovian paper, we don't see any correlation with a magnesium calcite ratio, for example. So it's non-dependent on that. And for cadmium, there's also theoretical work being done. The incorporation, the kinetic fractionation into the carbonates is not depending on the magnesium composite or the magnesium concentration in the carbonates. So for cadmium, I can definitely say this doesn't play a major role, but for nickel, barium, we have especially this works.
What's carrying last question? Before.
Yeah, please.
One question. So for cadmium, to go back to the cadmium question. So this type of process of precipitation by cells of cadmium, conditional sulfide, and the sulfide and particles.
Yeah, to store them actually in the cell, outer layers of the cell.
Yeah, and this happens also under oxy conditions, on it's really competition. Yeah, so I'm wondering if you believe that this kind of work that would be big enough so that eventually that impact your development scale, that would impact the
It's possible, yeah. And I mean, if you use the calculum as a bioproductivity proxy, it only will show you that there was enough material there to perform these processes. But yet, there is, it has been shown only once experimentally with E. coli, I think. And I think it's a very interesting concept because the actual fractionation behavior is the same. So it also favors light isotopes then, making the outer cell being lighter than the uptaken cadmium into the cell.
But we don't have any clue about the quantitative importance of this process in the biophysics.
Not yet, no. But a lot of these magnetotactic bacteria, for example, they do this kind of behavior as well, and that could eventually cause these actual metal isotope fractionation. It's very, yeah, still at the beginning to fully understand what's actually happening when this uptake is happening. Is that actually a nutrient uptake? Is it a real utilization, or as you say, detoxification because there's an overabundance of certain elements?
Okay, with Simon. You want to tell this neighbor you reduce stuff to the collection and like this. Merci pour les gens projets parce que merci à tous d'être venus. C'est vrai que yeah, we have now